Hi there. So I hope that before you are listening to me talk here, that you've watched the 3 links that I posted that have different advertisements for Pepsi in 1983. If we were in class today, physically together, one of the things that I like to do in class at this point is to actually do the Pepsi challenge that was depicted in those 1983 advertisements. That was a part of a big ad campaign that Pepsi did, and they suggested that there was some sort of a blind taste test, and they're showing you information about the results of that. And so what I wanted to have you think about is what sorts of things, when you see the Pepsi challenge, what do you need to think about in terms of actually designing a real experiment? Hopefully you can think of some things that maybe happen behind the scenes to allow Pepsi to create those commercials. So are they giving you a full data set? You're right, they're not. They're just cherry picking certain responses. So even though they're presenting this as we did this experiment, this blind taste test where people didn't know if they were testing, if they were tasting our product or our competitors, even though they're depicting that, they're not telling you what the actual results are. They're just cherry picking data. And so it's really important to remember that although they did a few things right, although they showed in the video that the design of their experiment was a blind taste test, they had product X and product Y, and they didn't tell anyone what it was supposedly. There are other things that maybe they didn't do so very well. And that's an important thing to bear in mind in terms of experiment design. So something that you might wonder about is, okay, if we're interested in what's more popular, Coke or Pepsi, we might just want to go to the store, right? Why can't I, in my time that I have on my hands these days, decide to just go camp out at the Jewel and keep a little tally? I can sit there in the soda aisle and just say, okay, I'm going to write down Coke and I'm going to write down Pepsi and other, and then I'm just going to tally. Oh, here's someone who bought some Coke. Here's someone who bought some Pepsi. Here are a few more people buying Coke. Someone bought some other kind of cola, maybe Dr. Pepper or something, and then Pepsi is bought, and then Coke is bought some more. Why don't I just do that if I'm interested? And the reason is experiments allow us to study specific treatments and to control for other lurking variables. So let's write that down together. Hopefully you have your notes packet out. I've written here that we should have our notes packet out to page 19, or you can just take your own notes, that's fine too. But the reason that we need experiments is because experiments allow us to control for specific things, and they also allow us to study the effects of specific treatments. Oops, what just happened there with my pen, sorry. We also can look at combined effects of combinations of factors. So a common example of that is to think about looking at how plants grow. And we know that for plants to grow, they need light and they need water. Well, I can take and make different groupings then, where plants get a variety of different amounts of light and a variety of different levels of water, and I can look for the best combination of those factors in terms of plant growth. So I can look at, can study combined effects of several factors. Okay, so to formalize this, there actually are 3 principles of experimental design. The first one is control, the second one is randomization, and the third one is replication. gosh, that list I just said sounds awfully like it could be a test question. The test question would be, what are the principles of experimental design? And you would say, control, randomization, replication. Let's talk about each of those in turn to make sure we know what they are. So control, you might have heard of this in terms of a control group. So a control group exists in an experiment because you're trying to remove the impact of lurking variables. So your control group is as similar as possible to your experimental group, except for the thing that you're trying to test. So if I wanted to see if a certain pair of socks was more comfortable, I might have everyone in my study put a normal pair of socks on their right foot and a new kind of sock on their left foot, and then compare those two. And the reason that would be a reasonably good study is that I am keeping everything the same about those 2 feet, except for the fact of the socks, and also being right foot and left foot, I guess. But I am walking around and my right foot and my left foot get used the same amount. I get hot or cold and my feet get sweaty or chilly in the same manner. And so it's really just the sock that would be different. And so I have a control group of my regular socked foot and I'm an experimental group of my foot that has the new sock on it. And I'm holding everything else being the same. And so that's what control is, holding everything else the same so that lurking variables are not factors. You're only testing the thing you think you're testing. Comparison is what we do in an experiment at the end. So this is an outline. You should definitely get used to drawing little like pictures like this or diagrams like this of particular studies. So a common question that you'll have over the next week or so is I'll give you like a paragraph and I want you to create a picture like this. So you'd say, okay, I need to have these 2 different groups and one group does one thing and the other group does the other thing. So like group A is going to take vitamin C, group B is going to take a pill that they don't know what it is, but it's like a placebo is what it's called. We'll talk about that in a minute. And then at the end, we're going to compare who has how many colds that season to see if the vitamin C changed how often people got the common cold. So that is what we call, that element of control is called comparison. And it allows us to know what has happened. And if we are controlling for everything else, comparison actually tells us about the thing that we want to study. Now, the diagram I just uncovered looks at a glance a little bit more complicated, but also kind of the same. It is kind of the same. The thing that's a little bit different at the beginning is that this is a diagram that shows block design. So just like in the diagram above it, you have a particular group getting treatment 1. This group is also getting treatment 1, but notice that of the 2 different groups that are getting treatment 1, half of them are over age 40 and half of them are under age 40. And then the people getting treatment 2, again, half of them are over age 40 and half of them are under age 40. I would use this kind of design in my experiment if I thought that age was important, right? And so sometimes it doesn't matter how old you are. But in a particular study, maybe something about what I'm studying will possibly interact with older people differently than younger people. And so I would want to include that in my design. So it might remind you of stratified sampling. Remember, in stratified sampling, you separate out the different elements, different experimental units into subgroups, and then you pick from the subgroups. This is similar to that, except it's for experimental design, right? I have my volunteers. I have an equal number of volunteers in each of the blocks, and then I split the blocks into however many groups I need. So in my diagram, I had 2 groups for each age grouping, but you might be testing like 3 different medications or 3 different kinds of footwear or whatever. And so then you might need to have more groups than that within each block. Okay, I just did this one as an example. Then at the end, you would still be using this element of comparison, right, at the very end. Okay, the second principle of experimental design is randomization. The point of randomization is to help prevent any kind of bias due to how you might select things just on your own. So the actual definition would be using chance to assign experimental units to treatments. I don't want to have some kind of favoritism, right? If I have awareness in a medical study of this drug is the actual drug and this drug is the placebo, I don't want to hand pick who gets what. My bias would come through in that. And so I want to use a random numbers table or draw names from a hat or use a computer numerical system, right? Like the random integer on my calculator or something along those lines to decide who ends up in which group. That is going to help me reduce that bias that would be present if I just did it in some other way. The third principle of experimental design is replication. And I personally always think of the ill-done study years ago that led people to think that autism and vaccines were linked. They are linked in that both of those things appear around age 2 for a child, but that doesn't mean, like those two things happening at the same time, doesn't mean that they are cause and effect. And the way that it was determined that that study was flawed was that there were other labs that tried to repeat it. So replication is a really important factor in experimental design. It is when you try to repeat your experiment yourself, just with more participants, just do it again. But even better is when it is done in a different lab or with a different researcher leading it to see if those results are correct. So when you add more participants and just repeat the experiment yourself, that is to reduce just variation that might happen by chance. But if someone else is repeating it, they are checking your results and making sure that you actually don't have a flaw in your data. All right, so rather than learning the particular names in this class of different kinds of sampling that can happen, or different kinds of experiments, I should say, I misspoke there, we could indeed try to learn different kinds of experimental designs. There's something called randomized experimental design, there's randomized block designs, there's factorial designs, there's all sorts of different actual designs. And I mention that because some of you might choose to take a stats class in college, and those definitely would be part of a college stats class. But what's more important for our class, which is just decidedly a little more basic than a college course, is to understand that all well-designed experiments have control, randomization, and replication. Those three elements definitely are the guiding principles of good experimental design. So here are some bullet points that all good experiments have. They make sure that they have random assignment to form groups as similar as possible before they apply the treatment. Okay, so the random assignment is that randomization, and they are trying to get the groups as similar as possible because that is what it means to have control over those lurking variables. They also, good experiments will use comparison at the end to see what is happening, comparison to make sure that everything is the same to start with, and then comparison at the end to separate out the effect of whatever treatments were imposed in the group. This is really the key idea about experiments. Okay, so let's say that you get to the end of your experiment and you're doing your comparison. Okay, and you want to compare. Remember the example I made up about plants, right? And so you want to see how watering and providing sunlight impacts the growth of plants. And at the end, you have this one corn plant that's gigantic, and you have some other corn plants that are kind of small. And you say, oh, it must be because this corn plant had the optimal amount of light and the optimal amount of water. Well, that's true as long as these groups were as similar as possible to start with, and as long as there's only a very small probability that it could have been due to chance. So let's look at this sentence. This is a really good sentence to take your time reading and understand. Differences that you have at the end of your experiment, differences that you observe between the groups are either just by chance, like coincidence, or they are because of the treatment that you imposed. Now, we don't really want it to be because of chance. We're hoping that it's because of the treatment, because that's how we answer questions. We want that big corn plant to be big because of something we did, not just because of happenstance. And so to minimize how much variation is due to chance, we have to make sure that we design our experiments really, really well so that the variation has to be minimal, because our group started out so much alike at the beginning. We had virtual twins at the start, except for the things that we were checking, okay? And that's going to help us be more confident that our results are because of the treatment. We actually can't prove it. We can only be more confident or less confident, and we want to be as confident as possible. So some other vocabulary that's going to come up within this study, some of these I think you probably know. The first one is the placebo effect, and people usually have some awareness of this. So a placebo is when someone is given a treatment that isn't actually a treatment. It just is fake. And this is because sometimes our brains can convince us that something should happen. So this happens sometimes, people will say, oh, I think I have fewer headaches when I wear this one kind of metal bracelet or something like that. And we can convince ourselves that because we think something will change, that something actually did change. That is just a susceptibility we have as human beings. So when we do a study, we don't want to compare not taking a pill to taking a pill, because the people not taking a pill clearly know that they aren't. So we want to compare taking a new medication that is real to taking a new medication that's just made of sugar, and then see what difference there is, because both groups will think that something should happen, and then we'll see if something different really does happen. Okay, a double-blind study. I had students last year who were really confused by this. A double-blind study is when the participants in the study don't know what they're getting. So you saw that in the Pepsi challenge commercials, right? People were trying soda X and soda Y, and they didn't know which one was which. Now, that's single blind. If the person who's interacting with that person also doesn't know which one is X and which one is Y, like the actual names of it, then that's called double blind. So for a double blind study, you need to have someone behind the scenes who's not interacting with the person in the study, who sets things up. So in a medication study, there'll be, drug A and drug B, and they're just put into containers by someone who knows what A and B mean. They're handed off to someone, and that person is the only person who interacts with the participants. And that person also doesn't know what A and B are, so they can't give anything away in terms of their attitude to then bias the data. A matched pairs design is actually what I was talking about when I said you're going to put a certain kind of sock on one of your feet, like your regular sock, and then a experimental sock on the other foot. Now, if you didn't know which sock was which, you were just told, put these socks on your right foot, put these socks on your left foot, and they visually looked the same, that would be a matched pairs design, because your feet match, right? They belong to the same human, and you could see if the treatment made a difference, right? Treatment 1 versus treatment 2. Matched pairs design is also used a lot with twins, because twins are as alike as possible genetically. And so you can give one treatment to one twin and one treatment to another twin. Matched pairs is also used with people who aren't twins necessarily. And I might say for the month of March, you're going to do this treatment. And for the month of April, you're going to do this other treatment. And then we'll compare how the 2 months went. And you'd have to have some amount of lack of awareness of what the 2 treatments exactly were in order for this to work exactly, because otherwise your own opinions would bias your data. And then a statistically significant result is a really hard thing to explain. I don't know why I had an arrow there. Maybe I just made that by accident. So a statistically significant result is when your observed effect is so large that it is unlikely to have occurred by chance. Okay, so an observed effect, ignore that arrow, so large that it is unlikely to have occurred or have happened by chance. Okay, and that's what I meant before when I said the differences between the groups either have to be due to chance or due to the treatment. When we think it's very unlikely to have occurred by chance, then we have what is called a statistically significant result because we're pretty sure our result is because of the treatment. So thank you so much for watching this video and for paying attention to those old commercials from 1983. You should ask your folks if they remember those commercials. I know I do. I was, a young teenager at that time. So there are some problems for you to do in your packet. And so make sure that you reference Schoology so you know your assignment. Thanks.